So we're working on two sorts of donor cells. So the first is we're working on fetal cells. So this is trying to transplant developing striatal cells taken from fetal brain. So, and we acquire fetal brain from terminations of pregnancy. So these, obviously with permission from the maternal donor and with full ethical permissions. So that's work that we've been doing for a long time, and there's a lot of evidence that fetal cells can repair the striatum in Huntington's animal models. And there have been some clinical studies. In fact, we undertook a study ourselves quite a lot of years ago now with, it was a small safety study. We are now about to start a new human study of fetal cell transplants in Huntington's, and that's using slightly higher doses of cells than have been used previously. So one of the problems that we think has been apparent from the data that we've got so far in humans is that it looks like the survival of the grafts hasn't been optimal in all cases. And we think that certainly in our last trial, we transplanted numbers of cells that were probably suboptimal. So that's something that we are now launching. That's going to be a very small study to start with, but we think that fetal cell transplants provide very important proof of concept studies for this for the cell therapy approach. In parallel to that, we and a lot of people globally are working on trying to derive donor cells from stem cell sources. So I'm talking about mostly pluripotent sources, so either human ES cells or human iPS cells. So we've got a parallel program that's been running for a number of years now. We have differentiation protocols, so we can make medium spiny neuron-like cells from human pluripotent cells. And some of the big questions we're working on at the moment is asking just how close are those cells to the authentic medium spiny neurons that we can derive from the fetal brain.